Stereochemistry is an important topic in the discovery and development of drugs, and it is gaining attention in the medical industry. The effectiveness of common drugs, such as ibuprofen and Prozac, are closely related to the stereochemistry of the molecules. Stereochemistry is defined as the study of the relative spatial arrangement of atoms that form the structure of molecules and their manipulation. In other words, stereochemistry is the study of isomers. Isomers are compounds with the same chemical formula but different arrangement of atoms. Isomers can be divided into two different categories: constitutional isomers and stereoisomers. Constitutional isomers have the same molecular formula but different connectivity. As you can see in the chloropropane example, the molecular formula for each molecule is identical, C3H7O. However, the connectivity is different. For one chloropropane, the chlorine is connected to the first carbon. For 2-chloropropane, the chlorine is connected to the middle carbon. Hence, they are constitutional isomers. Stereoisomers have the same connectivity but different spatial arrangement. Looking at the dimethyl cyclohexane example, these molecules are exactly the same except one is cis and the other is trans. For the cis isomer, the functional groups are on the same side of the carbon chain. Both methyl groups are on the wedge, meaning they are both sticking out of the screen towards you. For the trans isomer, one methyl group is on the wedge, while the other is on a dashed line. This means that one is sticking out towards you, while the other is going into the screen. Since these molecules have different spatial arrangements, they are stereoisomers. All stereoisomers have chiral centers. One type of chiral center can be identified by a tetrahedral carbon bonded to 4 different groups. A chiral molecule has the potential to have 2 different forms that are non-superimposable mirror images. A chiral molecule cannot have a plane of symmetry. Additionally, there are 2 different types of stereoisomers: enantiomers and diastereomers. Enantiomers are the mirror images of each other and have identical physical properties. Diastereomers are not mirror images and they have different physical properties. For a molecule to be a diastereomer, it must have at least 2 chiral centers. Enantiomers are usually present in a 50-50 ratio, known as a racemic mixture. To differentiate between the 2 enantiomers and a racemic mixture, they are assigned an R or S configuration. This visual can help with your understanding of enantiomers and diastereomers. The enantiomeric pair on the right and on the left are both mirror images of each other. The configuration is swapped at both chirality centers. However, each enantiomeric pair is a diastereomer to the other. If you compare molecule 1 to molecule 3, the chiral center with the bromine attached maintains the same configuration. It is not a mirror image. However, the chiral centers with the chlorine attached are mirror images. Since one chiral center changed, but the other did not, these molecules are diastereomers, not enantiomers. Now that we understand what enantiomers are, let's talk about them in the context of biological systems. As said before, enantiomers have nearly identical physical and chemical properties. However, enantiomers can behave very different in a biological setting due to chiral selectivity. A pair of enantiomers can affect proteins, enzymes, and metabolism in different ways. Therefore, a drug that is developed as a racemic mixture can have different effects based on the 2 different enantiomers. Some racemic drugs have one functional enantiomer and one inactive enantiomer. However, it is possible to convert a racemic mixture into the active enantiomer via chiral switching. Other racemic drugs have enantiomers that have 2 completely different functions. The enantiomers of chiral molecules can have different pharmacodynamic and pharmacokinetic properties. Pharmacokinetic properties involve absorption, distribution, biotransformation, and excretion. Chiral drugs are a relevant topic because they make up 40 to 50% of the pharmaceutical market. Of that 40 to 50%, 90% are a pair of enantiomers. Chiral drugs have generally been developed as mixtures of enantiomers because racemic mixtures can be difficult to separate. As technology continues to advance, more single enantiomer drugs may be seen. One example of a chiral drug is ibuprofen. Ibuprofen is usually sold as a racemic mixture, but only the S enantiomer is effective at relieving headaches. The body can convert the R enantiomer into the S, but this actually slows down the reaction time. Prozac is another example of a chiral drug. Prozac contains the molecule fluoxetine. The R enantiomer of fluoxetine is an effective antidepressant but has no effect against migraines. The S enantiomer of fluoxetine is effective at relieving migraines. As you can see, these two enantiomers have different medical effects. The topic of chiral drugs is very intriguing because though a pair of enantiomers have identical physical properties, they can have different effects in a biological system. Often a pair of enantiomers compete in a biological system. As technology advances, we can expect to see more beneficial single enantiomer drugs. As of today, over half of the pharmaceutical market consists of chiral drugs, and that number is expected to rise. For these reasons, stereochemistry is an essential topic in the pharmaceutical world.